Hello, everybody, and welcome to Meet a Scientist, sponsored by New Mexico EPSCoR. I'm your host, Anthony Salvagno from Explora Science Center in Albuquerque, New Mexico. Today, we have Dr. Bryce Chackerian, who is a vaccine researcher in the molecular genetics and microbiology department at UNM. Thanks for joining us today, Bryce.
Hi, Anthony. Thanks for having me.
So tell us a little bit about being a vaccine researcher.
Yeah, so I find it really exciting to work on vaccines. There are lots of diseases that we don't have good interventions for. So infectious diseases like viruses or bacteria that we're exposed to and can make us sick. But also lots of other things that can cause, real problems. So things like heart disease and Alzheimer's disease, chronic diseases. And so my lab is interested in trying to develop vaccines for all sorts of different things that can affect us. We're working on developing vaccines for bacterial infections, for viral infections, and then these chronic diseases that I mentioned. So things that affect the brain, things that affect the heart, and cancer as well. And all the vaccines that we make are based on a similar sort of technology. So what we've discovered is that our immune system, this is the thing that protects us from infections, our immune system is really good at responding to virus particles. Okay, so virus particles, when you think of a virus, I think a lot of you have probably done science fair projects where you've made virus particles out of styrofoam or out of Tinker Toys or something like that. And when you think about a virus particle, you think of a geometric shape, like a polygon or something like that. And it turns out the immune system, when it sees these shapes, it responds really strongly to it. It sees the geometry of a virus particle and it makes a really strong response to it. And so what we've done is we've taken advantage of that strong response to these shapes by taking a virus particle. And what we do is we use a virus particle that's completely safe. It doesn't infect humans. And we just use the outer shell of that virus-like particle. And then we display things that we want to target, that we want to make vaccines for on the outside of these particles. And so essentially what we're doing is we're tricking the immune system into responding to something by displaying it on the surface of something that looks like it's dangerous, but isn't dangerous. I think someone recently described this as sort of the reverse of the Trojan horse. So the Trojan horse looks safe on the outside, right? It's just a big wooden horse, but then it's full of soldiers from Athens on the inside, right? Ours is the exact opposite. It looks dangerous on the outside, okay, because it looks like a virus particle, but it's totally safe because it has absolutely nothing on the inside. And so we use that technology to develop vaccines against all sorts of different things.
So is the idea here that you're using, your own body to protect itself by introducing some, this virus looking thing, and your body will then just make everything it needs to do to be able to fight infection?
Yeah, that's absolutely the idea about how vaccines work. So, obviously, if you get infected with a virus or bacteria, it's bad news. And in most cases, your immune system can cope with that infection. But in certain cases, with certain things that are bad, you might get sick, and sometimes you can get really sick. So the idea behind our vaccines is that you're preparing the body to respond more quickly to a virus or a bacterial infection. So you have antibodies are something that your body makes to protect you. So you have antibodies that are already in your system, and so they're there and they can attach to the pathogen and protect you. And then there are other cells that these vaccines can also induce that allow you to respond more quickly. And so the idea is that you are already protected, and if you do get infected or if you do see an infection, you're able to respond quickly and you're protected from that.
I love the analogy of the Trojan horse. And, this is probably not true, but I have this picture now of you guys making tiny little horses that you are injecting into, and testing out in this process. What kinds of geometries do you use, and how do you even make those?
Yeah, so we actually use a very, very simple virus called a bacteriophage. So bacteriophages are viruses that infect bacteria. And it turns out that they're really, really common. So like your body actually has all sorts of bacteriophage inside you, in your gut, that are infecting the bacteria that live inside you. And they're all over the place. They're in the soil, they're in the ocean, there are probably some floating in the air. And again, because they only infect bacteria, they're totally harmless to us. And so what we've done is we've taken these very, very simple viruses, and then we figured out ways that we can manipulate them to stick things on the outside. So they don't exactly look like horses, but they look like little balls. And again, they have that sort of geometry that the immune system can respond really strongly to. And so we take these balls and then we use all sorts of different techniques. We use chemistry, we use biology to put things onto the outside. And that's one of the fun things about my job, is to think about what we're trying to target, what the disease is, try to figure out ways to stick things onto the outside of our virus particles. And again, there's various ways that we can do that. And then we test them, and sometimes they work, sometimes they don't work, but we just keep plugging away and try different things.
So I have to ask, I think a lot of people have seen this like electron microscope image of the COVID-19 virus, which is like the spherical thing with like little things sticking off of it. Have you guys tested your method on the COVID-19 virus?
Yeah, so those little things that are protruding from the surface of the COVID virus, these are spikes. Okay, it's called the spike protein of the virus, and it turns out that's a really important part of the virus for getting into cells. Okay, so when the virus infects you, it uses that spike protein as a way to sort of get inside of a cell, and that's how the virus is able to replicate. And so what we're doing actually is we're taking little pieces of that spike protein. Okay, and we're sticking it onto our vaccine platform system as a way to try to make immune responses against that protein, okay, against the spike protein. And so the idea is that our vaccine will then make those antibodies, make your immune system ready, so that if you do get infected with coronavirus, the virus won't be able to infect cells. At least that's the idea.
So if you, let's say you guys are able to, or anybody is able to get a vaccine for COVID-19 using this technology, like what would that look like for me to get the vaccine? Like how does that work internally?
Yeah, so just like most other vaccines, you'd get an injection, probably into your arm, okay, and it would be these particles. And vaccines are pretty safe generally, but sometimes you might get a little swelling at the site of injection. And then what happens is that the immune system, there's all sorts of cells in the immune system that can recognize these particles, and the immune system basically will say, "Hey, this looks dangerous. We need to respond to it." We start making antibodies. We start amplifying other sets of cells that can respond to it. And one of the consequences is a vaccination, that now you'll have antibodies that are inside your blood and hopefully circulating for a really, really long time. And so, again, if you do see the COVID virus, this coronavirus, then you have these antibodies that are there and ready. They're ready to go, and they're ready to block the virus. And so, that's basically how vaccines work, is they prepare your body so that you can respond really, really quickly if you happen to see the pathogen that you've been vaccinated against.
And that's so interesting. And I guess for all of the future scientists that are out there, how would somebody get into this line of work? Like, what did it take for little Bryce to get into immunology and vaccine research?
Yeah, I think it was a long road. I remember when I was in elementary school in fourth grade, I had to do my first science project. And I decided to do a science project on screws, which doesn't make any sense at all, because I didn't do an experiment, and I had a board, and I glued a bunch of screws onto the board. And I think my teacher was nice. She didn't say it was the worst science project that she had ever seen, but it was probably the worst science project that she had ever seen in her life. So anyway, so I've come a long way from then. I had some great biology teachers when I was in high school. I had really great math teachers when I was in high school. And that's when I started getting interested in science. I felt like the best thing about science is that there's so much that isn't known. um, you know, and I think the idea of being able to discover things, that's what's really inspiring to me. Doing something that nobody has ever done before, that's really, really exciting. So I finished high school, I went to college, I went to graduate school, that was 6 years. And then I did what's called a postdoc, and that was 7 years. All that training basically prepared me to take a job at the University of New Mexico. And it was a fun road. Got to do a lot of great science, did a lot of experiments. Most of those experiments didn't work because that's what science is about, trying stuff, and sometimes it doesn't work. But on the other hand, you do those experiments, you're able to put up with things that don't work for the experiments that do work, and those successes are really exciting.
And how long have you been at UNM since your
Yeah,
post?
so yeah, sorry, I've been here since 2004, so I've been here a little over 15 years. Yeah.
That's amazing. So what advice would you give your 10-year-old self to encourage them to stay on this path and to stick with it, for 6 plus 7 plus 15 years, plus, college and high school?
Yeah, I think the advice is to just stay curious. Well, first of all, get over this weird obsession with screws. That was probably not a good idea. But stay curious. Keep asking questions. Ask your teachers. Like if you don't understand something, ask your teachers. Teachers love to talk to you about all the things that they've learned throughout their career. And asking questions is a great way to sort of engage them and also engage yourself. So that's really my advice is, to just stay curious, ask questions, and think.
Thank you so much for participating in Meet a Scientist. Is there anything that you'd like to plug or share with everybody before we sign off?
Yeah, I just, vaccines are really important. I think that's my plug. That people worry sometimes about the side effects of vaccines, and there are a lot of people who are really against vaccines. But vaccines are really, really important. The reason that we don't have a lot of diseases that used to be common, like polio or measles, is because vaccines work really, really well. And I think that's also going to be true for COVID. But it's an important part of our healthcare system, and so I encourage you to get vaccinated and to learn more about vaccines as well.
So we'll all look forward to the Chackerian lab COVID-19 uh, vaccine. Thank you so much for doing this.
Thanks, Anthony. It was great.
Have a great day.
You too.